Also, we did make content slides. So if you guys wanted to review some things, we'll send that out too. But we're not gonna go over that today. But yeah. I'll actually, could you share the screen for me
Yeah.
instead? Okay. Okay, so question one. Okay, so these are like practice questions that we got from Dr. Cho, and they're pretty similar to what you'll see on the exam. Um, I'm pretty sure we'll be sending out like announcement with the recording. Yeah. Okay, so question one, it's asking about which ion is furthest from equilibrium. So we're gonna have you guys try this out first and like give you a few minutes to answer this before we go over like how to do this. So you guys can like start working on question one. Okay, so someone said B. Um, the answer is actually A. You can click next. Okay, and then you can also click again. Okay, so when you see a question like this that's set up like this with like the concentrations both inside and outside the cell, you're and since it's like also asking about like ions and equilibrium, your first thought should be like the Nernst potential equation. Because the Nernst potential, it measures like what voltage the ion what voltage the membrane needs to be for the ion to be at equilibrium. But so basically you would find the Nernst potential for each ion in the diagram. So I kind of have like the work for that out. So for X, the Nernst potential will be 61 millivolts. For Z plus, it's negative 82.5 millivolts, and for Y minus, it's negative 58.2 millivolts. And then once you have that, you need to compare it to the membrane potential. When you're looking at membrane potential, look at the number that's inside the cell. You're going to look at from an inside perspective. So since the membrane potential is negative 80 millivolts, you want to find the ion that's the furthest from that number, from -80. Don't look at like +80, look at -80. So since Z plus and Y minus, both of their nurse potentials are pretty close to -80, and X is the farthest from -80. So in this case, X would be the ion that's the farthest from equilibrium. And then conversely, you could say that like the Z plus ion is the closest to equilibrium. So yeah, in questions like this, once you have like the nurse potential of the ion, it's like telling you exactly like what value the membrane potential needs to go to for the ion to be at equilibrium. If you wanted the X ion to be at equilibrium, you would need to find like a way to get -80 millivolts down up to 61 millivolts. So yeah, that's question one. Okay. Um, so we'll give you a few minutes to do both of these. And if you have like an idea what the answer is, you can just like drop it in the chat. But yeah. Okay. So you did get the answer for 14 right, but 13's actually A. Yeah. Okay. Okay. You can click it 2 more times. Okay. So 13, it is kind of confusing, but because like you would think that because there's a greater concentration on the outside, it would want to flow in. But this is also like a nursed equation question. So first you would calculate the nurse potential for Y. And so you can see it's 42.6 millivolts. And so that at that value is where Y is going to be in equilibrium. If the membrane potential was at 42.6, there would be no net diffusion of Y. I mean, like the ions would still be moving, but they wouldn't be moving in like one like overall direction. So usually like for things to whenever like something isn't in equilibrium, it's always trying to move towards equilibrium. So since the membrane potential is at 61 millivolts, um Y would want it to be at 42.6 millivolts. So for that to happen, the memory potential needs to drop to 42.6 millivolts. And for that to happen, it would need to lose its positive charge. Since Y is a positively charged ion, when it leaves the cell, it leaves behind a negative charge, and that's going to drop the membrane potential. So it would end up diffusing out of the cell. So basically, like the Y ion would leave the cell so that it can get the membrane potential to drop to its equilibrium potential. Um, so and then once it hits 42.6 millivolts, the membrane potential, the ion wouldn't be like having a specific direction that it would move in. It would just be like like zero net diffusion. And then 14 is just asking like it's just 14 is like more simple. It's just asking like what is the membrane potential required for it to be in equilibrium, and that's basically like the definition for the Nernst potential. It's just the memory potential that tells you like it just tells you the memory potential an ion needs to be in equilibrium. So for this one, it's just like a simple calculation of the Nernst potential, and that's how you get D.
All right, so next, all of these questions are referring to figure 2, so I'll give you guys like a minute or two to do these. I just made a Zoom poll that you guys can put your questions in or your answers in if you want. I'll give you guys like 30 more seconds. All right, so let's look at number 3. Most of you guys picked D, and let's look at the answer. So yes, D is correct. Number 2, you guys picked or for number 4. Number 4, you guys picked C the most. It was actually E. Okay. 5, we have you guys picked B, all got it right. 4, you guys all got it right. So figure 2, which point is sodium furthest from equilibrium? So this question kind of goes into back into what Suba was talking about. So the Nernst equation is the equation that tells you what voltage do you need for equilibrium to be reached, right? So if we actually knew the concentration, so this is an example of an axon. If we actually knew the numbers for sodium on the left and the right, and we also knew potassium, whatever, we would have been able to figure out what voltage we would need for sodium to stop diffusing. So if you actually figure that out, like with numbers, you'll the Nernst equilibrium voltage for sodium is somewhere around 40, okay? It's like 40 or a little bit higher than that. And if you do the same for potassium, you'll find that its equilibrium voltage is around negative 65. And then here, as you can see, I wrote each phase is driven by the highest permeable ion. So knowing that, the question says, where is sodium furthest from equilibrium? We know at equilibrium for sodium is around 40, somewhere up here. So whichever part of the line is farthest from that, it's going to be D. So that's why number 3 is D. All right. And then 4, people also had a little conflict with what they picked there. So in figure 2, which position are all voltage gated channels closed? I have another slide that explains more specifically. Let's see. So 5, everyone got right, basically. You use the same concept as number 3. And then number 6, you guys all got right. So I'm going to go into this next slide here. So at this table will summarize for you what happens at each phase. At the resting phase, the only thing that's open are your potassium leak channels. Obviously, to maintain this potential, you also need the sodium potassium ATP pump. You should recall that. However, we're talking about channels here, not pumps. So the only thing open is the leak pump, which because it's most permeable, it kind of drags our membrane voltage down to negative 65 and it stays there. When a stimulus causes a depolarization to around negative 50 millivolts, right? So right here, that's our voltage threshold. What happens is 2 channels will open, okay? The sodium one opens, and then the potassium one slowly opens, okay? Slowly. So as you can see here, we're in the rising phase. Sodium is open, but the potassium one, it's delayed. So anyway, so rising phase, because potassium gates open, we're gonna be going towards the sodium, equilibrium voltage. So that's why we're going up there. All right, so after we reach 40 millivolts, the voltage gate closes, okay? For sodium closes. And then finally, our voltage gated potassium channels are fully open at this. So because of that, once again, sodium or potassium is now the most permeable. So it's going to drag our voltage downwards, okay? So that was the falling phase. In the undershoot phase, the only thing you need to know is that the sodium or the voltage gated potassium channel takes some extra time to close as well. So it takes time for it to open and it also takes time for it to close. So because of that, we actually allow we hyperpolarize, and then it's called undershoot as well. And then eventually we just flatten out again after we go all the way back up to the resting phase. Okay, so that explains um, figure 2 here. So basically, only at the resting phase only the leaky channel is open, and everywhere else you actually have a voltage gated channel open. Okay, so now for number 11, it's asking more about the inactivation gate. In the discussion, someone mentioned or asked to explain more about how the inactivation gate and the activation gate affect these graphs. So take some time to do this question. Okay, I also opened another poll. And also as a hint, the sodium gate has two parts to it, the activation gate and then the inactivation gate. Okay, so almost everyone already put an answer in the poll. The answer is actually E, okay? So let's talk about why. So first, let's just go through how this thing works. So the activation gate opens at -50, our voltage threshold, okay? And it closes at our voltage threshold. So think back to or we actually have one right here. So here we have some depolarization. We reach the threshold, the activation gate opens, okay? Which allows sodium to rush in, increasing the potential. And then, as you can see, it doesn't close again until it reaches the threshold. So what closes it? What allows this thing to go down? Well, obviously potassium flowing out allows it to go down, right? But also the inactivation gate closes at the peak here, which is finally what stops sodium from flowing in. Okay. And then remember I said the hyperpolarization part of the curve was due to the potassium one taking a little bit longer. Um, so yeah, this peak here will not change, or this dip here won't change simply because we're not affecting the voltage gated potassium one. We're only affecting the voltage gated sodium one, all right? So basically you can cross out any graph that doesn't have this hyperpolarization undershoot. So that's why we could eliminate choice C. The undershoot is caused by the voltage gated potassium one, not the not the the sodium one, okay? Something else you should understand is there's a refractory period. If you remember one of those slides in Dr. Cho's lecture, there's an absolute refractory period, which goes from like here to here, and then there's a relative refractory period. The fact that the sodium voltage gated channel only can open again at -70, which is below your resting state, explains the absolute refractory period. Basically, once we are in this state here, you cannot fire another action potential because the inactivation gate will only open once you reach, once the voltage is dragged down a little bit farther than it's at resting, okay? That's what explains that absolute refractory period. That might show up on the exam. And so now let's look at all of our choices. A lot of you guys picked A, most of you guys picked A. And um, so you guys picked it because the thing is higher. So what you should understand is we're delaying the inactivation gate, okay? And if you remember I said earlier in the last question, the Nernst potential for sodium is around 40 or just a little bit higher. And so in reality, it wouldn't actually be able to reach this high because of that fact, and also because we did not change the potassium gate at all. So you would actually see some like plateauing here. So basically, the flow of sodium in and the flow of potassium out, they're kind of like fighting each other. So you actually see more of this very sharp curve here. Okay, so that's why we would eliminate choice A. You want to see one of these. And then, like I said earlier, we also eliminate C simply because C is illustrating a change in the voltage gated potassium, but we're not changing that at all. We're only changing the sodium one. So that's why E is the correct choice here. As we reach here, the delay in opening, right, makes it so it's just fighting the flow of potassium. So that's why you kind of see this, plateau here instead of the straight drop off. And then here, we should still see that undershoot.
Okay, so for 1516, we'll give you some time to answer that too. Um, and there should be a poll for 15 and 16. So try just answering through that. Okay, so most people put B for 15 and A for 16. And those are the right answers. You can click. And then yeah, okay. So okay. So for 15, it's asking about increasing intracellular potassium. So with this one, you can look at like the fact that potassium is kind of associated with the resting, falling, and recovery phases. And so during the resting phase, yeah, during the resting phase, potassium is kind of like what determines what the resting potential is. So during that phase, potassium is trying to leave the cell, and it's making the inner membrane potential more negative. So if there's way more potassium inside the cell, then there's like an overall greater amount that's trying to leave the cell. And so that's going to result in the membrane potential becoming even more negative. So with B, it's kind of the fact that like the resting potential area is lower than -65. And that's kind of what lets you know like that the potassium is getting like the potassium concentration is affecting that. When it comes to like the voltages changing in these graphs, it's usually because of like the concentration of an ion changing. So that's kind of like the similar idea for 16. 16 is A because when you look at the peak of graph A, it's way above 40. Um, and that's because like with the overshoot part, when you're during the rising phase, the sodium channels, the voltage gated sodium channels are open. And so this is where sodium is flowing into the cell. So if there's a lot more sodium outside the cell, then a lot more sodium is going to flow in. And this is what causes like the membrane potential at the peak to be even more positive or like greater or higher than 40 millivolts. So that is kind of how you would answer 15 and 16. But I'll kind of go over the other graphs a little bit. So with um A, it's kind of like you can see how it's different from the control because of like the peak changing, and that would be because of the increased extracellular sodium concentration. B, it was the threshold, like the resting phase in the very beginning of the graph is way below -65, and that would be like the cause as a result of like a lot of potassium leaving the cell. For C, um graph C, after the overshoot at the peak, during the falling phase, you can see that the falling phase is occurring like very slowly. Um, and you can see it's like a very sharp decline. So that would like indicate that there's something wrong with the potassium channels because it is that's kind of where like potassium it like takes dominance in that part of the action potential, the falling phase. So when you see a sharp decline like this, it's like a sharp decline, but it's still happening very slowly. That means the repolarization is happening very slowly. So this kind of just means that there's like a problem with the potassium channels. Something is causing it, um, causing the potassium to leak out of the cell slower than usual. So that's like the most important part of C, just like that part of the graph. With E, it's also showing that it's taking with, oh wait, yeah, with the E, it's also showing that it's taking a while for the repolarization to occur. But the reason why it's taking a while is because of like that little plateau region that's right at the overshoot. And I think he just talked about this. That area is kind of like when you think of the overshoot, you think of the sodium inactivation gate closing. And so if there's a problem with the inactivation gate, then that would mean like that part would happen much slower. For the repolarization to happen, you would need the inactivation gate and the potassium channels, the inactivation gate to close and the potassium channels to open. But since this starting point right here is happening like really slowly, you can just like pinpoint that it's probably in the inactivation gate that's causing like a delay in the inactivation gate that's causing this to happen. So those are like the most important parts that you should like understand about these graphs when you're answering the questions during the exam. Um, yeah. Okay.
Okay, so I'm gonna be going over questions 7 through 10. I don't know if somebody could make a poll for this, I think
I just
one of the
opened one.
TAs. Okay, sorry. Um, but yeah, I guess we'll just hang out and wait for the poll results. We have like half of the people who have participated so far, so I'll just give it like 1 or 2 more minutes. You guys can get that submitted. Just 2 more people. Okay, I'm gonna give it like 10 more seconds and then I'll close the poll. Okay. So let me share the results. So we're gonna go through one by one. So first for number 7, so like A and B have an excitatory effect of 10 millivolts and C has an inhibitory of 10 millivolts. And then D inhibits B, but that isn't actually relevant to this question. It has like a lot of information at the beginning that can confuse you. But when you actually get to the core of the question, what would you expect to measure on the voltmeter if A and C spike at the same time? Well, A has an excitatory effect of 10 millivolts and C has an inhibitory effect of 10 millivolts. So basically they're just going to cancel each other out and there's going to be no change. And then number 8 is A. So again, it's like the same little blurb of information to start the question. But then the actual question is, what would you expect to measure if D, B, and C all spiked at once? Well, C has an inhibitory effect of 10 millivolts. And then D inhibits B by 5 millivolts. So B is excitatory, but D inhibits a little bit of that excitation. So think of it kind of as like B is positive 10, and then D comes in and inhibits 5 of those millivolts. So B is actually only like a positive 5 millivolts. And then C is negative 10 millivolts. So if you sum that all together, it's going to still be negative 5 millivolts, and that's inhibitory. And then number 9 is C. So what would you measure if D were to spike 5 times? Well, if it's just D spiking, D only has an impact on B. So if B isn't really doing anything, then neither is D to the actual potential. And then for 10, the answer is D. So what transmitter is likely to be released by A? Well, A is an excitatory potential of like positive 10 millivolts. And so I'm just gonna like go through each answer choice and explain why D is right. So AMPA is a receptor, it's not a transmitter, so A is automatically out. GABA or glycine, they're inhibitory and A is excitatory, so that one's out. Dopamine can be excitatory, but most of these synapses use amino acid transmitters, and glutamate is a common neurotransmitter for excitatory potentials. And then so that's why D is the right answer, because it's the most common excitatory neurotransmitter, and A is excitatory. Any questions?
I could probably share my screen for this part.
All right.
Can everyone see my screen?
It looks good.
Cool. So basically, I will be talking about figure 10. There was actually a lot of questions regarding figure 10 and 9. But before I get into practice questions, I just want to provide like a general overview of this chart. This isn't the exact chart that you'll see on the exam, but it's pretty similar. This is actually from the lecture. We could start off by going over the somatic nervous system. So one thing to note here is that there's not going to be a 2 neuron series with the somatic nervous system. So what I mean by that is that there's just going to be one long axon that directly connects the central nervous system to the effector organ. And the effector organ in this case would be our skeletal muscle, right? So essentially signals are being directly transmitted from our central nervous system to our effector organ. And this is going to be mediated by motor neurons. In terms of preganglionic neurons and postganglionic neurons, this was also another question that I saw on the discussion board. So basically, what preganglionic neurons is that they're directly going to originate from the central nervous system, right? So they're going to be a set of neurons and fibers that originate from the central nervous system, and they're going to transmit signals to a target neuron, right? They're going to transmit signals to a target neuron through the release of neurotransmitters. So a general rule of thumb that you should remember in terms of preganglionic neurons is that anything that directly originates from the CNS or the central nervous system is going to release a neurotransmitter known as acetylcholine, right? And this neurotransmitter acetylcholine will bind to nicotinic receptors. So the general rule, just remember anything with preganglionic neurons is going to release acetylcholine, which binds to nicotinic receptors. This is found in all cases in the somatic nervous system because it's directly connected to the central nervous system. And it's found in both in the sympathetic and parasympathetic. But you're going to see a lot more deviations right here in the bottom half. But I'd like to go over postganglionic neurons first. So in terms of postganglionic neurons, when they're depolarized after binding of this neurotransmitter, what they're going to do is that they're going to transmit this signal or impulse, and essentially this action potential causes the release of these different sets of neurotransmitters that bind to effector organ. So postganglionic neurons, a signal transmits and is essentially sent to an effector organ. So in terms of the parasympathetic nervous system, when I think of parasympathetic, I think of rest and digest. So just know that with this, it's associated with the muscarinic receptors. When I think muscarinic, I think mushrooms, and that's essentially associated with relaxation. And essentially, this is mediated by the binding of acetylcholine. In terms of sympathetic nervous system, this is going to be a little bit different because essentially neurotransmitters can be essentially released by different mechanisms in this case. For this first case, this is going to be normal synaptic transmission, right? So here, an action potential is essentially spread through this postganglionic neuron, and this causes the release of norepinephrine. What happens to this norepinephrine is that it binds to adrenergic receptors. Adrenergic is just another way of saying adrenaline receptors. And sympathetic, remember, is fight or flight. So fight or flight, adrenaline. So that's an easy way to remember sympathetic, as well as the receptor that is associated with it. But there's another mechanism for the sympathetic nervous system, and this is associated with the adrenal medulla. And this is basically a gland that secretes hormones. So when it's activated by the binding of acetylcholine to this nicotinic receptor, an action potential is fired within this adrenal gland, and this causes the secretion of hormones into our bloodstream. This hormone is known as epinephrine. And just like what I said with this normal synaptic transmission, this neurotransmitter is going to bind to an adrenaline receptor, right, and cause a fight or flight response. So you guys should really be familiar with this chart in terms of the exam, because he's going to ask a lot of like questions about ligand or receptor pairs and stuff like that. So now we're going to move on to the practice questions. Okay. I'll give you guys around maybe like a minute or so. I'll probably give you guys maybe 30 more seconds. All right. So if no one else is going to answer, I'll go over the answers right now. So basically for 17, the answer is actually going to be A. So, I'm sorry. Yeah. Okay. Yeah. So question 17 is asking which ligand receptor pair is going to use slow metabotropic receptors directly. So you guys should be familiar with that one chart in terms of where it lists all the neurotransmitters down. So basically what you should remember is that when acetylcholine binds to muscarinic receptors, it's going to cause this signal transduction pathway, like through the GPCRs and stuff like that, which is example of a metabotropic effect. So when acetylcholine binds to muscarinic, it's an example of a metabotropic effect. And another case is when norepinephrine binds to its receptor. Norepinephrine binding essentially is another example of a metabotropic receptor. And in this case, on this chart, we can see that acetylcholine or the muscarinic receptors is going to be listed out as number 3. And when norepinephrine binds to adrenergic receptors, that's going to cause metabotropic effects, which is listed out as 4, 5, and 6. So for this case, the answer is A. And in terms of 19, the question's asking about endocrine signaling. When you think endocrine, the first thing that should come to mind is hormones, right? So essentially with hormones, the only thing associated with it is actually 6, right? In terms of 6, the binding of acetylcholine to this nicotinic receptor on the adrenal medulla causes the secretion of epinephrine, right? And epinephrine is actually going to travel through our bloodstream to our effector organs to cause the fight or flight response. So in terms of endocrine signaling, it's going to be 6. It's not necessarily 2 and 6 because 2 is not endocrine, it's through synapses, through synaptic transmission. So in this case, it's just 6 for endocrine. Moving on to the next question. I'll give you guys maybe a minute and a half for these. This one's a little tricky. Give you 30 more seconds. All right, so for question 19, it's asking which ligand receptor pair in figure 10 directly requires snare proteins to function. Okay, so with snare proteins, remember what snare proteins are actually responsible for. Oh, what the heck just happened? Sorry, 1 second. Okay, yeah. Remember the function of snare proteins is to essentially regulate vesicle fusion with the membrane so that you can release neurotransmitters. And essentially, since all of these are related to the release of neurotransmitters, and if we don't have snare proteins, essentially now we can't release neurotransmitters and therefore we cannot transmit signals throughout our body. So therefore, in this case, which ligand receptor pair, it's going to be all pairs. In terms of 20, the question's asking which ligand receptor pair includes sites that directly signal with norepinephrine, right? So when I think norepinephrine, I think the sympathetic nervous system, right? Specifically in this bottom half right here, because in this first half, remember when it's derived directly from the central nervous system, the preganglionic neuron is just going to release acetylcholine, which binds to nicotinic receptors only. That's the general rule of thumb. But right here, we're referencing this bottom half. So sympathetic nervous system is going to initiate the fight or flight response by releasing norepinephrine. And essentially, norepinephrine binds directly to these adrenergic receptors. One thing that I did forget to mention, though, is that the adrenal medulla doesn't just send out or secrete epinephrine. It can also secrete norepinephrine to initiate this fight or flight response. So in this case, which sites directly signal with norepinephrine? It would be 4, 5, and 6. All right. Okay. Should have a slide here. Okay, there it is. Okay, so for this one, I'm just going to be talking about different scenarios that can essentially correlate with this information flow. This is something that he'll definitely test you on in the exam. I'm just going to provide some general examples that could probably help you a little bit. But before that, I'm going to provide a general overview of this figure since there are a lot of questions associated with it. So generally, information is going to be, or like outside information is going to be processed in our body. Our receptors detect this information. And essentially what they do is they send this directly to the brain and the central nervous system through what is called the afferent division. So if you're transmitting signals to the central nervous system, that's what is known as the afferent division. And what happens is that interneurons in the brain and the spinal cord are essentially going to process that information. So how I like to think about it is that they are these mini processing centers within the central nervous system that are going to make decisions on what physiological response to carry out if necessary. So if they do come up with some decision in terms of what response to carry out, they're going to send out this instruction manual through this efferent division. Efferent, E for exit. And essentially the signal is going to branch out in either the autonomic nervous system or the somatic nervous system. So essentially, if you want to carry out actions that involve the movement of our skeletal muscles, that's going to involve the somatic nervous system and control over muscles through motor neurons, right? Whereas if it travels through the autonomic nervous system, this impulse, when I think autonomic, I think automatic, right? These are things that are not going to be under our control, right? They just happen. Like we can't control our heart and just say it should be faster, or we can't control our gut to say, you know, digest your food a little bit quicker or something like that. In this case, the autonomic nervous system can be divided in both the sympathetic and parasympathetic nervous system, right? So remember, sympathetic is associated with fight or flight. And when I think of parasympathetic, I think rest and digest. In terms of this enteric nervous system stuff, this is not going to be tested, so you shouldn't really worry about this branch. So one last thing I'd like to mention is that autonomic nervous system is going to regulate these set of effector organs, right? It's going to regulate the smooth muscle located in your gut or your digestive system, your cardiac muscle to control your heart rate, exocrine glands, which can relate to sweat or, saliva secretion, and endocrine glands, so like the secretion of hormones. So an example for scenario A, where essentially the signal is traveling from the sensory stimuli, afferent division, the brain, and eventually to our skeletal muscle, would be picking up your spoon to eat your bowl of cereal, right? So essentially the stimuli in this case would be maybe like seeing the spoon, and essentially that the receptors in our eyes detect that information, send it out to our brain, and our brain processes this information. What it does is send instructions directly to our somatic nervous system to instruct our arms to pick up the spoon. So that would be a good example of scenario A. In the case of B, essentially the information is directly traveling from this sensory stimuli all the way to our sympathetic nervous system, right? So when I think sympathetic nervous system, once again, I think fight or flight, right? So in this case, a good example would be increasing heart rate. Heart rate is something we cannot control. So essentially when we see a physiology exam, typically we start to panic, right? So this information, once we see that exam, are detected by our receptors, sent out to our brain, and this essentially instructs or sends out an impulse directly to our cardiac muscle to essentially beat faster during these stressful situations, right? So this is a good example. The complete opposite would be scenario C, where essentially information is traveling all the way to the parasympathetic nervous system. So when I think parasympathetic, I think rest and digest, right? So once again, because we cannot control our heart rate, this is going to be under autonomic control. These things occur automatically, right? For case D, information is, okay, so the big difference between case D and E is that information is not going to be it's not going to be derived externally, right? So we're not going to have a sensory stimuli that essentially sends information to our brain to carry out a response. Instead, all of these impulses, these signals are originating within our brain. So when I think of when it originates from the brain, I typically think of dreams or nightmares, as you might have seen in your homework assignments. So essentially, because scenario D talks about controlling our skeletal muscles, essentially by the impulse is being derived from our brain and spinal cord. An example I said was probably boxing Mike Tyson in your dream and punching the air while sleeping. So because this is not going to be derived from an external stimuli, all our thoughts and processes that are occurring on our brain is essentially going to send out instructions to our arm muscles to essentially start punching the air, but this is all occurring subconsciously. We're not aware of it, right? In terms of E, 3, 4, 6, 7, so information is transferring directly to the sympathetic nervous system. An example would be pupil dilation during a nightmare. When I think pupil dilation, that's one of the physiological responses associated with the fight or flight response, right? And because it's a nightmare, it's going to be derived straight from your brain and spinal cord, right? Not externally. These are all occurring in your thoughts and in your process or nightmares that is essentially causing you to feel like you're in a life-threatening situation. And essentially what it does is that your brain will send out these instructions to your sympathetic nervous system and to control essentially your pupils, right? So that's it for that. This is just a general overview of things that you might see during your exam, like the physiological responses associated with the sympathetic, as well as your parasympathetic nervous system. So one thing you should look out for is heartbeats. That's a common one. Saliva secretion, dilation of pupils. Another thing would be digestion, and yeah. So just get a general understanding of what physiological responses is associated with each system. All right, so Ka, if you want to share your screen again, that would be great. Oh.
Okay, I'll go over this question. So these are questions. We have like a few things that we didn't go over in the practice questions that were from the discussion that we'll just go over right now. This kind of relates to figure 5, but this person was asking about what would happen if there's like a delay in the activation gate. So and they're asking like why the answer wouldn't be C, why it's B instead. So C is similar to what you saw in figure 5 for graph E. C is because of the inactivation gate having a delay. But since this question's about that is asking about the activation gate, it can't be C. It would have to do the activation gate, if there's like a problem with that, that's probably going to affect like the rising phase area of the graph instead. And so if like there's a delay in the activation gate, that means it's going to take a lot longer for the potential to like depolarize, because depolarization occurs because like the sodium starts flowing into the cell, which is raising the potential of the membrane. And the way sodium flows in is through its channel, but for its channel to be open, the activation gate has to be open as well. So if there's a delay in the activation gate of the sodium channel, it's going to be affecting the rising phase. Like it's going to be affecting the amount of time it takes to get to the rising phase. So with B, you can see that it takes about like 2 milliseconds to even get to the peak, and it takes about like 1 second to start rising. It takes like about an entire second for it to like complete the rising phase, but with the other graphs, it happens like almost like even quicker. So that's why the answer here would be B. And then with C, it's just like what we talked about before. C would be like a delay in the inactivation gate. And then I just wanted to mention one thing. So when the sodium channels are open and depolarization is occurring, so during like the rising phase, this is when the activation gate is open, but the inactivation gate is also open. That's how like the sodium is going through. But when you get to the peak at the overshoot, the activation gate will still be open, but the inactivation gate closes then, and that's how you get sodium to stop flowing into the cell. And then you can go to the next question. Oh wait, actually, could you go back? A is like So with A, you're seeing that like the peak is way above 40 millivolts. So that's just telling you that there's like a lot more sodium that's flowing into the cell. Basically like the change is that it's causing the depolarization to reach like a much higher potential than like the average at like 40 millivolts. So for that to happen, you could like increase the amount of extracellular sodium, the amount of sodium that's outside of the cell. If you increase that amount like by a lot, that's going to end up increasing the membrane potential. So A would be possible if you like increase extracellular sodium concentration. Okay, and then this is just like a figure I put in here that kind of explains like what happens, like which gates are open during each phase. So if you guys are confused about that, you guys can kind of take a look at this figure because I think it's pretty detailed. So, but yeah, it's just a supplemental figure for you guys to look at. Next question. Okay, so this question was about one of the clicker questions he went over. So okay, so which is true for the sodium gradients at the time indicated by the arrow. So the time indicated at the arrow, that's the overshoot phase. So by this point, there's been like a lot of influx of sodium into the cell. So and for the sodium to go into the cell, there needs to be a sodium concentration gradient that goes from like outside to the inside. So basically here you're looking at like, when you're looking at the concentration gradient, just think about like what you know is happening. When you're getting to the peak, the sodium is flowing into the cell. So the concentration of sodium is going to be inward. But when you get to the peak at this point, now that all the sodium is like, not all the sodium, but a lot of sodium is inside the cell. So the potential is really positive. And you can think of it as like it's very positively charged inside of the cell. Sodium is a positive ion. And since like charges repel each other, sodium would want to get away from positive charge to a negative charge. At the overshoot, you can think of it as like the inside membrane potential is positive and the outside is negative. So sodium would want to flow as an ion, it would want to flow out of the cell. So that's why the electrical gradient is out. Yeah, that's why the electrical gradient is out, like outward. And then so basically the positive ion would be attracted to the negative outside membrane. But yeah, that's why when you're looking at these questions, you should just think of them as like, try to think of what the situation is, like what's going on. You should think about like sodium is usually flowing in, but at this point as an ion, like think of it as like from the perspective of like the charges. As an ion, it's positive, so it's not going to want to stay in the positive region. And okay, and then also sodium is at its Nernst potential where equilibrium is reached.
Isabella?
Oh, sorry. I am gonna go ahead and share my screen for these next couple Sorry if my Wi-Fi starts to get laggy. My computer always starts to crash when I start sharing my screen. Okay, if somebody could unmute themselves and let me know if they see this. Okay, I'm gonna take that as a yes. Okay, so there was a few questions asking us to go over figure 13 and some of the questions on the homework associated with that. So, and people also asked me to go over AMPA channels and NMDA channels. So I will go over all of that like in this slide. So first I'm gonna go over what AMPA channels even are. So AMPA channels are glutamate-gated ion channels that mediate excitatory postsynaptic potentials. So glutamate will bind, and then that is what allows the ions to flow and cause the postsynaptic potential. And NMDA receptors, essentially they mediate long-term potentiation. And tetanus is a sorry, I'm getting a little sidetracked, but so like somebody asked why A was the control, I think, for tetanus. A is the control because tetanus is the long-lasting increase in amplitude of EPSP. And just to like go back on NMDA receptors, so NMDA is associated with long-term potentiation, and it has MG that blocks NMDA, and so like glutamate will bind to NMDA, but this won't remove the MG. The MG is only removed from NMDA when there's a strong depolarization and glutamate, and this will open NMDA and allow calcium to go through NMDA. And then this calcium stimulates the insertion of more AMPA channels, which allows the increase of the excitatory postsynaptic potentials, which ultimately helps create tetanus and long-term potentiation. So to summarize all of that, AMPA channels are always there, and they help create the EPSP. And then the NMDA receptors, they only open when there's a strong depolarization and glutamate, and they allow for more AMPA channels to be integrated into the postsynaptic neuron, so that way you can have a stronger EPSP because it allows for more AMPA channels. So if we were to block AMPA channels, what would you get? So this is the control, and then we block AMPA channels. You go to C, because if you don't have AMPA channels, you can't get like an EPSP. You can't get any potential, postsynaptic potential. So that's why it goes down to 0, which is C. And if you block NMDA receptors, your AMPA channels are still there. So you will still get that depolarization. You just won't get the long-term potentiation. So if you block the NMDA, if you block the NMDA receptors, you'll just continue to get this, which is B, because you won't get those extra AMPA channels integrated that allows for that increase in EPSP. And then, so when you add nicotine, you would get A, because nicotine is an agonist, which activates a receptor, and agonists usually excite sodium potassium channels. So if you add an agonist, you can stimulate more EPSP, and therefore you would get A, because if you stimulate more EPSP, then you get And then, so for this last question, what does tetrodotoxin, I don't think I'm pronouncing that right. So if you think about what that essentially does, it blocks sodium channels. So if you block that sodium channel, then you really can't get any EPSP. So that's why it goes down to C, because like I said, you're blocking those sodium channels. And if anybody like has any questions on figure 13 for the exam, or why like any of those things might happen, now might be a good time to ask about those. Let me see if I can see the chat.
I have a quick question.
Okay, like wait one
If
minute, if anybody has any questions.
if tetanus wasn't there, would adding nicotine still have the same effect?
If ketamine was there,
No, if like the tetanus wasn't there, would adding nicotine still have that effect, or would it be like B?
so I think it still would have that effect because nicotine is an agonist, and agonists will activate the receptor.
Okay, gotcha. It just like wouldn't be as strong. Like the tetanus makes it stronger.
What wouldn't be as strong?
Like the EPSP.
Yeah, I guess so. I guess you are right, and I see like where that line of reasoning is because the tetanus like allows for there to be more receptors.
For
Okay,
this
thank
test, I
you.
could just probably like add on. So, role of nicotine is to, as its name suggests, is to increase the activity of nicotinic receptors. So, in the postsynaptic membrane, you're not just going to have those AMPA receptors. You're also going to have those nicotinic receptors that allow sodium to flow in. So if you have nicotine, you're going to boost those nicotinic receptors. So you're going to allow more sodium to essentially flow into the cell. Therefore, you're going to have a stronger, what's it called, postsynaptic potential, which would exhibit like a relatively similar behavior to tetanus, if that makes any sense. Yeah.
Okay, yeah, thank you.
Okay. So that's figure 13. And then so this was figure 11, and there was also a lot of questions about figure 11. And this kind of this refers to like that one fish or slug or whatever it was that like when you would touch it, like at first it would withdraw, but like over time it stopped. And so that's what's kind of shown here is habituation, is that over time the withdrawal stops. And then like a painful stimuli was introduced, which kind of caused like a reset or the sensitization process, which then reset the withdrawal. But over time, habituation happens again and it happens faster. So the withdrawal will decrease again. And there was like a lot of questions relating to what could cause B. So the main thing that is changing in B is the fact that sensitization is not happening again. You don't really get that big jump back up to like what the original level of withdrawal was. So when you think of what could happen or like what would have to happen to remove sensitization, you could do that by knocking out the GPCR because GPCR helps with sensitization. And if you knock out GPCR, this doesn't have a big impact on like habituation in the first place. So this would still happen, like habituation would still happen. But if you were to knock it out, you wouldn't get that sensitization and you wouldn't get that big jump back to like what you were originally, which is like what happened with A. And then what could happen to like cause C? So what's happening in C is that you have no movement or withdrawal at all. So you really don't have any postsynaptic potential happening. And like when you think about what could happen to get no postsynaptic potential, you could do that by blocking the sodium ion channels. And so like if you block those sodium channels or you knock them out, then you can't get a postsynaptic potential. And if you can't get a postsynaptic potential, then you can't get this withdrawal. And something that could, or like a chemical that might be able to do that is, I know I'm not going to pronounce this right, but Curia or something like that, because it blocks the activation of skeletal muscles, and it blocks the activation of skeletal muscles by blocking nerve impulses. So if you block those nerve impulses, then you obviously can't get any withdrawal. So that's how you can get C, where you just don't have any movement at all. And is there any questions on this figure?
Also, I just wanted to add on. So another way of getting like C, where essentially like flat lines, is through Botox, right? So remember botulinum toxin. What it does is that it actually causes the degradation of snare proteins. And remember what snare proteins are responsible for? They're responsible for neurotransmitter release. And essentially, if you have no snare proteins, essentially these vesicles can't fuse with our membrane, and therefore you cannot release these neurotransmitters. And if there's no neurotransmitters in the synapse, there's essentially nothing binding to these receptors to cause ions to flow into the postsynaptic membrane. So if there's no ions flowing in, there's no graded potential, and essentially no sensitization in this case. So yeah.
I believe that's the end of what I have. So we can.
I actually had one more thing to add, but about like a figure before. Could you go back to slide 20?
I don't know the slide numbers, but like let me know when to stop.
It's the slide that starts with like, keep going. And then one more time. Yeah, wait, go back. This one. Okay. Oh, wait. No, wait, sorry. Keep going again.
Which way?
Forward. One more.
I can stop sharing if needed, like
Okay.
if you want to share.
So for this question, you can also think of it at like one more way. At the overshoot, sodium is at its nurse potential because its nurse potential is really close to 40 millivolts. So if it's at nurse potential, like if the membrane is at the nurse potential of sodium, that means sodium is in equilibrium. So for if that ion is in equilibrium, its electrochemical gradients should cancel out. Because you're supposed to have like no net diffusion. So that's also kind of another reason for why the concentration gradient and electrical gradient are in opposite directions in this question. But yeah, that's just like another way to look at it. That's all I had to mention.
All right, so if not, I think that's all we had. So we'll send out these slides, the recording, and we'll also send out like another supplemental
Hello.
thing as well. Oh, yeah, did you have a question?
Did y'all only do practice question, or do you guys do like like you guys used to do review slides? No
Just the
review
practice
slides?
questions. We'll send out the review slides as well. So it'll be separate, but we did not go over it.
Oh, no breakout rooms?
Hmm?
No breakout rooms?
Yeah, no
Oh.
breakout rooms, just the practice questions. Just so I think you saw everything.
Yeah, no breakout rooms, but if you have a question right now, you should ask it.
No, I needed time to freak out, but it's fine. That sucks. I'll blame all of you when I fail tomorrow.
You won't fail.
Trust me. This time it's not clicking at all. I don't get it.
All right, so if you don't have a question, thank you for coming, everyone.
Yeah, we'll
Thank-
hang out like
I
for
appreciate you.
1 more minute.
Thank you.
Thank you.
You're
I'm
welcome.
sorry, could I ask one more question relating to the last slide with the gill withdrawal?
Yeah, 1 sec. Let me
Sure.
go there.
Thank
Yeah.
you. Regarding CAM, I know that the like presynaptic synapse like starts CAMP and like the GPCR eventually leads to that being produced. So if you were to knock out or like not have any CAMP, would that be an example of B?
Wait, so if you were to knock out camp because it's like a second messenger of
Yeah.
GPCR,
Yes.
yeah, I think that that would have like the same effect because like if you don't have camp, then you really can't achieve the effect of the GPCR anyway.
Okay. Thank you so much.
Yeah, no problem.
Anyone?
Did anyone have any other questions?
Hi, Danae. You're alone now.